Thanks for joining us. We're here in Mission Control, and I'm joined by Dr. Vic Cooley, who's the lead increment scientist. Thanks for joining us this morning. So we're right in this great transition phase. We've got a crew that's preparing to depart. They've been wrapping up their long-duration mission, which has been heavily focused on science, and we're readying for a new crew. We know that you have a team, you're part of a team that works on strategically planning all of this science. Can you tell us a little bit about what Expedition 41 has done? What type of highlights there have been from their science?
Well, thank you, Nicole. I'd be glad to. Yes, so over the past 8 weeks or so, we've conducted Increment 41. It's been an extreme challenge because of two EVAs that were not planned during this time period, were added late just before the increment began. But despite that, and each EVA brings with it on the order of 100 to 150 hours of additional crew time activity to accommodate and prepare for and then clean up after those EVAs. But despite all that, we were able to get all of the critical science done. And perhaps the key accomplishment in the science activity, certainly a new one for the space station, is the enhanced rodent research facility. Now, we've flown rodents on the shuttle system over 20 times over a 30-year period of the shuttle life, but in this case, this is an expanded capability. We use rodents as models for bone and muscle loss primarily, and again, that's what this effort was about in Increment 41. There were two key objectives to prove that the new system, consisting of three pieces of hardware, the transporter unit, the habitat that the mice live in for the however many days they're in weightlessness, and an access unit, which is used to transfer mice between their habitat and a work volume. So all three of those pieces of hardware worked as expected, and the second objective was to return frozen tissue from the mice after either 20 days for some of the mice and 30 days for additional mice. That tissue has been returned from the 20-day mice on the SpaceX that landed in late October, so I should say splashed down off the coast of California on the SpaceX 4 mission. So the two main objectives for that research were well accomplished, and now we're looking forward to more research in that area.
And is there more, I guess you were alluding to, there's more of that experiment and research planned for Expedition 42?
Not for Expedition 42, but in Expedition 43 will be a SpaceX 6 mission, 2 SpaceX vehicles from now, and it will extend the challenge to the hardware system. We've proven that the hardware system can support mice and keep them healthy for 30 days, and now we're going to extend that to 60 days. Of course, it's designed to support rodents that long. So there's a big future for rodent research. In the 2011 decadal research by the National Institute of Health, or I'm getting my acronyms confused. It wasn't National Institute of Health, but it was one of the recommending agencies that helps NASA do long-term planning. And it's called a decadal survey. That survey strongly recommended that station develop a rodent research capability. So this is the first response to that, and I expect we'll be doing rodent research probably in an ongoing basis for years to come.
Okay, can you tell us some of the other research that is planned for Expedition 42?
I'd be glad to. The last time I talked, it was with Brandy about 2 months ago. We talked about the Increment 41 science, and that time I talked about model organisms in all the biological experiments that we had over the past 8 weeks. I want to do something similar today on the SpaceX 5 mission in December. We have, I want to talk about 7 experiments that are really biological in nature. And I want to do that by going through the 3 laboratory modules one at a time and talking about 2 or 3 investigations in each. So, but first let me point out that, these model organism biological type investigations are in no way the only type of science we have on the space station. We have lots of physical science in materials research and so forth. We have lots of research looking out at the rest of the solar system and the universe, and we have lots of human adaptation research going on to allow us to explore the solar system. But today, and because of the SpaceX 5 mission, which is particularly well suited for biological type experiments, which characteristically have to go up for the nominal mission is 30 days and then come home after that period of time. That profile is well suited to these biological missions, so that's why I talked about them for SpaceX 4, and that's why I'm talking about them again for SpaceX 5. So starting in the Columbus module, we have 2 cellular level experiments and 1 plant experiment. Yes, so the Columbus module shown on the left there is known as the, well, it's the ESA, European Space Agency laboratory known as Columbus. The first experiment I want to talk about is called Triple Lux. This uses blue muscle hemocytes. Now, hemocytes are phagocytes in this case. They actually detect and ingest foreign bodies. So this would be a immune response in the plasma of the blood, so to speak. It's not a cell-mediated immune response. It's outside the other cells, and these hemocytes are dedicated. Their sole purpose in life is to detect and ingest and eliminate invading threats to the organism. So in this case, the hemocytes will be incubated, some at 0G and some at 1G, and we use the 1G as a control to make sure that we're not misinterpreting the effects of weightlessness with the effects of radiation, because that's the other strong difference between on the Earth and in space, is the radiation environment is quite a bit different. So many life science samples are affected by radiation. So several of our experiments have a 0G and a 1G version of them. This experiment has that. We will look through a microscope with video at these hemocytes as they ingest foreign material, both in 1G and 0G. So that's the Triple Lux experiment. The second experiment in the Columbus module is T-cell activation in aging. This one uses human blood cells, human white blood cells known as T cells. T for the thymus. The human thymus is below the sternum, and this is where those cells are produced. These cells have special receptors on their surface that respond to threats, and they trigger immune reactions in other cells. So it's these T cells will be incubated in the cubic incubator in the ESA module, and after 75 hours they will be fixed and returned to Earth, and we will do some genetic analysis on them there. So that's a little bit different from the first experiment, the Triple Lux, where we were talking about dedicated cells like antibodies and antigens that go after and eliminate threats. In this case, it's a cell that it merely detects and then triggers other cellular responses to respond to the immune system. So there's a fundamental difference there. In the first case, it was invertebrate cells of the blue muscle, the aquatic blue muscle, not muscle cells, but MUSSEL in this case. And then in the second case, it's actually human white blood cells. So the third experiment I want to talk about is a plant experiment, and you did speak about the seedling growth experiment that's going on this week. Another plant experiment in the ESA module is APEX 3. This is Advanced Plant Experiment, and this experiment looks at plant cells. In particular, we're growing Arabidopsis seeds, which happen to be the same seeds in the current experiment, but this approach is fundamentally different in that the plant cell walls have structural elements known as microfilaments and tubin or actin, which are plant cell walls. Basically, all plant cells aid in the structure of holding the plant up against gravity, unlike human cells, or unlike animal cells. So in plant cells, we are again using biomolecular genetics at the very smallest level that we're at today in our forefront of science, and studying how mechanisms signal each other throughout the organism. So we're looking at signaling pathways for how the plants change and respond differently in 0G versus 1G, because now they don't have to hold up their structure. They don't need as much of these actin filaments in the cell walls, and they grow differently. So we're looking at that. That experiment actually returns plant tissue fixed with paraformaldehyde, which allows the cell structure to remain intact, but we also fix other samples with a fixative called RNA later, and those are frozen, unlike the paraformaldehyde samples, which can only be refrigerated so the freezing won't burst the cell walls. For the genetic RNA later fixative, those cells are frozen, and then we can do molecular analysis on the frozen samples. But a third result from this plant experiment is that towards the end of the experiment, after several days of growth, the plant tissue will be taken into the US lab where we have a fluorescent microscope, and many of the proteins, some of the proteins that are key in signaling the actin, how strong to grow, have been tagged with a green fluorescent protein, a so-called reporter gene, that allows an observer through on the space station through this fluorescent microscope, and it's actually a camera that's looking through the microscope, so the scientists on the ground can see the camera and record the video. And the green fluorescent protein is a technique developed in the 90s and well matured today, and it's widely used, so much so that we actually have 3 fluorescent microscopes on the space station. This one happens to be in the fluids rack in the US lab. So we have 3 ways of looking at the results: the refrigerated return, the frozen return, and the video return of the green fluorescent protein, which tags the structural elements. Yeah, you can see the US lab is shown circled there in the center. So 2 other experiments we have in the US lab as we're proceeding through the 3 labs now. The second experiment in the U.S. lab is known as Micro 5. This uses the model organism C. elegans, which is a roundworm. It's also known as the nematode. And this experiment looks at the combined effect of reduced immunity. We know that in humans and in all organisms, there is actually reduced immune response in microgravity. And we also know that for some pathogens and bacteria like Salmonella, there's increased virulence. So the combination of these 2 effects is both detrimental to the host, and we're looking at the interactions of increased virulence and decreased immunity in the case of these nematode cells. And I'm trying to remember, these are actual full organisms of the nematode, I'm pretty sure, in response to the Salmonella. So in this experiment, we'll again return both refrigerated and frozen samples and do cellular and molecular analysis on those.
Well, this sounds like a lot of fascinating science to come. I'm sorry, we're going to have to wrap it here, but
okay.
It just sounds like there's so much going on, lots of exciting research, and the best place for folks to learn more about these experiments is on our website at www.nasa.gov/station. So thank you so much, Dr. Cooley, for joining us, and we look forward to following along with this.
I'd love to come back. Thank you.